Okay, well, here we go. So I've realized that I've been going over this Pro-Opiomelanocortin protein many times before. So it's called POMC or POMC, apparently is how it's referred to by people who work in the field. Anyways, this is a protein that is synthesized and is broken down into its constituent peptide hormones, and they are stored and await release given particular cellular signaling. So here I'm going to just show in, well, you know what, it doesn't have a photo here, and I do not want to look up a copywritten photo, but just know that it is synthesized in the hypothalamus, and then it gets proteolytically cleaved and processed, and then gets transported into the pituitary. And you know what, rather than just explain it, I will go through this review called POMC, the physiological power of hormone processing. So I'd like to thank these people for creating such a really thought out review. So Pro-Opiomelanocortin is the archetypal polypeptide precursor of hormones and neuropeptides. So in this review, we examine the variability in the individual peptides produced in different tissues and the impact of the simultaneous presence of their precursors or fragments. So there is a lot of variability. Well, think about it this way. Let's say you produce your protein with a bunch of different peptides that are within it. And then these can circulate throughout the entire body. So if you are an intelligent organism, you are going to use that protein in many different ways depending on what cell type or what type of tissue those proteins are a part of. And this can be an excellent way of getting multiple signals out of one singular protein. Now, one could argue that it's dangerous to have so many needs hinging on one particular protein, but we could also assume or presume that there is a lot of redundancy and robustness in having one protein serve multiple functions, and then we could also assume that there will be some robustness among what parts of that protein. Oh, how to phrase that? If some parts of it fail, we can assume that there will be negative feedback loops that will hopefully self-regulate the entire process. So let's continue. We also discussed the problems inherent in accurately measuring which of the precursors and their derived peptides are present in biological samples. So here's another issue, is that this one protein actually has many different peptides in it, and a lot of the amino acid sequences overlap within those peptides. So then it becomes extremely difficult to try to figure out which peptide is associated with which physiological role, and when we are looking into the tissues to figure out what peptides are in it, we sometimes have a difficult time knowing which peptides are physiologically relevant, which peptides are just the waste products. Yeah, it ends up becoming, biology is a noisy field, so it becomes progressively more difficult to separate variables, especially when all those variables are being packed together into one protein. So continuing, we address how not being able to measure all the combinations of precursors and fragments quantitatively has affected our understanding of the pathophysiology associated with POMC processing, or POMC. So here they're acknowledging our weaknesses is going to make it more difficult to understand the mechanisms of the pathophysiology. So in a lot of what we do for a lot of genetic diseases and metabolic diseases is that we try to figure out the mechanism by which those diseases arise. So this comes from understanding what the role of a particular protein or an enzyme is, And then we figure out, okay, if this is too weak or too strong, then therefore it causes X, Y, and Z in the metabolism, and then that can result in the pathology of a disease. So by understanding the mechanisms, by understanding how the proteins work together, we can then gain a comprehensive understanding, or a more comprehensive understanding, of the disease in question. So here they're saying that since this particular protein is so intertwined with a lot of different things in the body and is intertwined with basically its processing by proteases, then that will make it much more difficult to untangle all the roles that POMC has. So, continuing, to understand how different ratios of peptides arise, we describe the role of the prohormone convertase, or the PCs, or prohormone convertases, and their tissue specificities, and consider the cellular processing pathways which enable regulated secretion of different peptides, and these peptides that play a crucial role in integrating a range of vital physiological functions. So this is a run-on sentence. It's a sentence as long as a paragraph. So we'll break this down for you. So to understand how different ratios of peptides arise, this POMC protein, when it gets cleaved into multiple peptides, then each of those peptides may or may not have an important role in whatever tissue it's in. And so the ratios of all these peptide sequences could be what would the word be? Could be important, or we would argue is important, since it's evolutionarily conserved to some degree. We would argue that it is important in whatever function, in the functioning of the tissues. And so what can cause those ratio differences? Well, those ratio differences will be predominantly dictated by the prohormone convertases. So these will basically be the peptidases, or the proteases, that will find a part on the POMC molecule or the constituent peptides and break them up further. So if you could imagine, you have your POMC, it gets broken up into two important peptides, but let's say you only want one of those peptides to still be active in that particular cell. Well, that one cell might have an additional prohormone convertase, which then deactivates that other peptide. So if you could imagine, your cellular tissue will have a set of prohormone convertases that it normally expresses, and then those prohormone convertases will dictate which peptides remain in their active form. Okay? And this could be how one hormone can do multiple things in different tissues. So I guess what I'm trying to get at is tissues are specialized, and one of the ways that they can become specialized or remain specialized in their function is to handle hormones differently, and one of the ways to do that is using prohormone convertases. So these are called PCs. And their tissue specificities, consider the cellular processing pathways, and then this enables regulated secretion of different peptides that play crucial roles in integrating a range of vital physiological functions. So that's basically what I just said. So in the pituitary, correct processing of POMC peptides is essential to maintain the hypothalamic pituitary adrenal axis, or the HPA axis is also what it is called. So a lot of proteins are synthesized in the hypothalamus, they get processed and moved into the pituitary, and then they are released from the pituitary, and a lot of those released peptides will then act upon the adrenal cortex, or the adrenal tissues, and that is another way in which more hormones can be produced. but we'll talk more about this later. And this processing can be disrupted in POMC expressing tumors. So if you have a tumor that unfortunately has the mechanisms or the biosynthetic pathways to produce POMC, that can disrupt your hormones. So for instance, if you know what, I don't know if if it's exactly correct, but I'm remembering a story of I believe he was European giant, and he basically had a tumor in his pituitary gland, and that tumor ended up causing a huge release of human growth hormone throughout his developmental periods, and he ended up being a giant. And it was actually a matter of huge concern because that could have killed him later on. I don't remember how it was resolved, but it was resolved. But if you get tumors in these, and we're making the assumption that the tumor is that the tumor is in one of these important secretory or what would the word be, hormonally relevant areas, that if you get a tumor there, the tumor, like the original tissue, is going to be expressing different hormone proteins or different hormones. So if you have a tumor, let's say in the hypothalamus, you might actually start producing way too much POMC. And if you produce way too much POMC, then you might produce all of the subsequent peptides in concentrations that are way too high for your body. And you could have pathophysiology, or you could have pathology that results from that. And let's continue. In the hypothalamic neurons expressing POMC or POMC, abnormalities in processing critically impact on the regulation of appetite, energy, homeostasis, and body composition. So if you, in your hypothalamic neurons, because the hypothalamus is a part of the brain, they express POMC, and if they do not do that properly, then you have a much more difficult time regulating appetite, energy, homeostasis, and your basically the components of your body. So your musculature, I'd assume, and your adipose tissue as well, your fat. Continuing, more work is needed to understand whether expression of POMC gene in a tissue equates to release of bioactive peptides. So this is one thing. Perhaps certain cells express POMC or will produce POMC, but that doesn't necessarily mean that the POMC is going to be processed into bioactive peptides. So, and perhaps there's always the off chance that POMC in of itself might have its own function as the full-fledged protein. So it's very difficult to rule out anything. It's almost as if the more you research any protein, the more you research any component of biology, then you find other roles that these proteins play, the more you dig into them, which is why basic research is so necessary to have a comprehensive understanding of nature. Continuing, we suggest that this comprehensive view of POMC processing, with a focus on gaining a better understanding of the combination of peptides produced and their relative bioactivity, is a necessity for all involved in studying this fascinating physiological regulatory phenomenon. And so, how do I phrase this? Everyone who does research on their particular protein very frequently will talk about how great their particular protein is, because the more you research and, I would say, come to love a particular component of physiology, you end up, I don't know, speaking more highly of this particular protein. Especially if your grant money is somewhat contingent or how do I phrase this? Especially if you receive grant money in order to, as a result of selling yourself and selling your research, that would be another contributing factor as to why people talk so highly of their research and the particular proteins that they research. So I've been rambling, and we are already 15 minutes in, so let's move forward. And I'm going to stop at about an hour because I think we should take this slowly. But I do believe that it is necessary to understand what POMC is before we understand how it gets attacked in viruses, or how viruses use peptides within POMC, or at least one. I believe it's more, but at least one peptide in POMC is being attacked for in order to increase pathogenicity and to increase symptomology so that the viruses are much more likely to spread. Anyways, the discovery of POMC as a precursor. So the phenomena of Pro-Opiomelanocortin, or POMC, as a hormone precursor emerged gradually over time as observations slowly filled in pieces of the puzzle. So long before the concept of hormone precursors was realized, the bronze skin color described by Addison in his patient with adrenal insufficiency, and he called it melasma suprarenale, he gave perhaps the first hints of a connection between the hypothalamic pituitary adrenal axis, or the HPA axis, and skin color. So keep in mind, Pro-Opiomelanocortin, melano is within it, right? Melanin. Melanin is one of the things that is affected by this pro-protein hormone. Okay, so let's move forward. A similar link between the pituitary and pigmentation came from the studies of Allen and Smith, who both noted that immersing tadpoles in pituitary extracts made their skin darker. In humans, too, large doses of porcine pituitary extract, so they probably took the pituitary glands from pigs. I would presume after they were slaughtered for meat, they probably had some contract with whatever meat processing facility, and they basically took a bunch of pituitary glands, blended them up, and had some used some extraction mechanism or some extracting solvent. And anyways, but if they did that and gave it to humans, it appeared to cause pigmentation, or oh, that's actually funny, pigmentation. That it's a pun right there. Sorry. So it ended up causing the skin to darken. With this active extract of the pars intermedia and of the pituitary, henceforth termed melanocyte stimulating hormone, or MSH. So I don't know what the pars intermedia is. So it's the boundary between the anterior and posterior lobes of the pituitary. So it contains 3 types of cells. Basically, it's a part of the pituitary that separates the front and the back. Continuing. Oh, and so they called this extract melanocyte stimulating hormone, or MSH. But what we know now is MSH is only a part or a piece of the whole puzzle. So continuing. In 1932, Cushing extended his clinical reports of a polyglandular syndrome caused by basophilic adenomas of the pituitary by linking this finding with adrenal hyperactivity. So, a lot of jargon there. Let's break it down. So, Cushing, he extended his clinical reports of a polyglandular syndrome. So, polyglandular, this is involving multiple glands or multiple hormone-releasing organs, if you will. And this syndrome was caused by basophilic adenoma. So what is a basophilic adenoma? It's a cancer, but let's see if I can get a Wikipedia. So pituitary, it would be a pituitary adenoma, and these are tumors that occur in the pituitary gland, and pituitary adenomas are generally divided into 3 categories dependent on their biological functioning. Benign adenoma, invasive adenoma, and carcinomas. So let's see, okay, so basophilic adenoma means that it is a cancer that likes a basic or a high pH, if I'm reading this correctly. So historic or histological classification utilizes an immunohistological characterization of the tumors in terms of their hormone production. So historically, these were classified as either basophilic, acidophilic, or chromophobic, on the basis of whether or not they took up the tincture stains, hematoxylin, and eosin. Oh, okay, sorry. I'm wrong. I'm sorry about that. It's not about how the cell lives in the organism. It's about how the cell, the cancer, how it stains in the microscope slide. So when we image a lot of these cancers, we will stain them with different types of stain, and certain cells actually will take stains at different rates. And so the basophilic one will take one particular type of dye. The acidophilic one will take a different type of dye. And then there's the chromophobic ones that have a very difficult time taking up any type of stain. So yeah, I apologize. That is what they mean by basophilic adenomas, is that they are cancers that are stained by a certain type of stains, and I assume that these are basic stains. So anyways, this polyglandular syndrome was caused by a basophilic cancer of the pituitary, and he extended his clinical reports by linking this finding with adrenal hyperactivity. So they were able to find a link, and the link was with adrenal hyperactivity. So in 1930s, work by Engel and Kendall showed that administration of large amounts of cortin, which will be important later, this is a purified adrenal extract. So think of the adrenal cortex, cortin. That's how they ended up naming this. A purified adrenal extract. This is this extract produced atrophy, or the basically the reduction of, it's basically as if the cells are dying or shrinking, so, or wasting away would be the best way to describe it. So this produced the wasting away of the adrenal cortex in rats. So importantly, they found that administration of the adrenotropic principle of the anterior pituitary was effective in preventing adrenal cortical regression following treatment with cortin. So there was something, and that something was called the adrenotropic principle, or think of adreno and tropic, or something that will make it thrive or make it grow. So this tropic principle is the adrenotropic principle. So it's helping the adrenal glands survive. And this was from the anterior, or the backside, or anterior pituitary, I should say. And this was effective at preventing the negative consequences of adding cortin, which, remember, the addition of cortin was causing the atrophy of the adrenal cortex. But with this adrenotropic principle from the anterior pituitary, that stopped that from happening. It stopped the atrophy. And so this is one of the ways that the pituitary ends up interacting with the adrenal cortex. So let's continue. The first hints of a behavioral angle of POMC or POMC biology came from studies by Ferrari in the 1950s, when stretching yawning syndrome, a bizarre crisis of muscular tone, occurred following central administration of melanocyte-stimulating hormone. So remember, this was a hormone which was associated with the production of melanocytes, or darkening the skin or causing pigmentation. Anyways, many other studies assess the effects of central alpha melanocyte stimulating hormone on motivational processes followed. But it was not until 1976, that Panskep observed for the first time that this peptide just decreased food intake. So I don't know if this was being done on humans or done on rats at this point, or done on some model organism. So, but I would say probably the way that it's pronounced, I would guess it was that. But let me look this up real quick. 294. So, effects of alpha melanocyte stimulating hormone on motivation, vigilance, and brain respiration. I don't know if I think that said rats. Yeah. So this was being done on rats, not on people. So let's not be. So if you could imagine, when these people were giving melanocyte-stimulating hormone, I would assume a purified extract, which was originally its use was for causing skin pigmentation. When they added it to these rats, they ended up getting stretching yawning syndrome, which is basically they started wasting away. They started being too tired, I would presume. Anyways, it was not until 1976 that Panskep observed for the first time that this peptide decreased food intake. So somehow this melanocyte-stimulating hormone is associated with food uptake. So that's strange, but I mean, this is a strange protein, right? So, viewed from the comfort and assured knowledge of the modern molecular world, these observations and interventions could be considered overtly simplistic. So, if these people had modern molecular technology at their disposal, all of this would have made sense. But to them, it was a puzzle piece that was very difficult to put together. So we could imagine it as they are, I forgot who said it, but now we get to stand on the shoulders of giants to have all of this information at our disposal, and we can come up with very effective models for the effects that we see. So, standing on the shoulders of giants. Who said that? Bernard of... This concept has been traced to the 12th century, attributed to Bernard of Chartres, or I don't know how to pronounce that. So, a French Neoplatonist philosopher. Okay. So, that is true. Oh, sorry. Where are we at? Oh, here we go. Nope, all the way here. So, what does that tell us? So this tells us that today, since we have all this technology, and we are able to take the information from our experiments, the conclusions from our experiments, and we can hypothesize, we can make models, and we are better at doing that now. I should say the models are much more complex and more robust now, because we were able to stand on the shoulders of giants and utilize all of the research that has been done before us. So, yeah, we should continue. So viewed from, so however, we believe that these classic observations should be regarded as essential building blocks, not only for our understanding of POMC, peptide processing, but also for the work which subsequently tied together these seemingly diverse peptides. So remember, these are all building blocks that get put together into its final model, or at least almost a final model now. So the emergence of the precursor paradigm. So it is likely that POMC arose over 500 million years ago by an insertion of the melanocortin sequence into a pre-proendorphin gene. That is interesting. I am going to save, that's 266. So evidence for this comes from the structural identities with other opioid precursors in both the N-terminal and C-terminal regions of POMC. So I'm actually very intrigued by that. So 266. Oh. So this is 60 years of POMC and evolutionary oh, wait. I believe, okay, that's a different one. So let's look, I apologize. So yeah, I'm definitely going to read this one later. So I'm going to deviate a little bit from that review for a second to just talk about this for a little bit. So these people are arguing with this, in this journal specifically, these people are arguing that POMC is a protein that has been around for 500 million years. Over 500 million years ago. So 500 million years. That is a long time for it to be in circulation. So what does this tell me? It tells me that it has been around for so long and is probably conserved in so many creatures today that it would be a good target molecule for viruses to abuse when they infect different species. So, and you could imagine that there is coevolution going on. Both the viruses and the organisms that have this POMC, they're both mutating in conjunction with each other. Interesting. I will go over that more, but let's continue with this. So evidence for this comes from structural identity. Oh yeah, I read this. So the common opioid gene was thought to arise during chordate evolution. So if you're not familiar, so these are what chordates are. Chordates form a phylum of animals that are defined by having, at some stage in their lives, all of the following anatomical features. So a notochord, which is a fairly stiff rod of cartilage that extends along the inside of the body, and among vertebrates, subgroups of chordates, the notochords develop into spines. So this notochord is turning into a spine in the vertebrates. Okay? So that's one of their features. The other features is a dorsal neural tube. So in fish and other vertebrates, this develops into the spinal cord, the main communication trunk of the nervous system. So we have a dorsal neural tube as well. Spine, so keep in mind the notochord turns into the spine, the neural tube turns into the spinal cord. So those are two important distinct features. And I should mention, it doesn't always turn into a spine, and it doesn't always turn into a spinal cord, but these are the features, these exist at some point in time. Also, there are pharyngeal slits, so it's a part of the throat immediately behind the mouth. And so these slits, you even have these at some point in your, as a fetus, you do have these slits as well. In fish, these slits will turn into gills, but for you, I don't know what ends up happening to them. And then they will all have a post-anal tail. So it's a muscular tail that extends backward behind the anus. So for instance, when you're a fetus, you will have a tail, but that tail ends up disappearing. And then also, all chordates have an endostyle, or an endostele, which is a groove in the ventricle wall of the pharynx. And for instance, in filter-feeding species, it produces mucus, which helps transport food to the esophagus. So this endostyle may be a precursor to the thyroid gland in humans. Now, keep in mind, these are all very difficult to see in all creatures, because you would have to monitor the entire life cycle of creatures to identify these things. But this is sort of the rough, I shouldn't say rough, but the well-refined but probably still incomplete definition of what a chordate animal is. So the common opioid gene has thought to arise during chordate evolution. So in this particular subset of animals, this is where the opioid gene probably arose. So a long time ago. So there are 4 opioid genes that are on 3 chromosomes in the vertebrate genome. An intragenic duplication event in tetrapods is thought to have led to the presence of alpha melanocyte stimulating hormone, beta melanocyte stimulating hormone, and gamma melanocyte stimulating hormone. The gamma melanocyte stimulating hormone sequence is not present in teleos. I forget what a teleost is, but a fish of a large group that comprises all ray-finned fish species. Okay, so it's a type of fish type thing. So a fish with a bone, I think, is what they're talking about. And it's found as a vestige in non-teleos, whereas an additional melanocortin peptide, termed delta melanocyte stimulating hormone, has been found in cartilaginous fish. This suggests a divergence in melanocyte stimulating hormone sequences in cartilaginous ray and lobe-finned fish. So this sentence is basically telling us that the opioid genes are ancient. And then sometime around 500 million years ago, there was a melanocortin sequence that got inserted into the preproendorphin gene. And remember, endorphins are opioid. They are opioids. So somehow that got inserted in very long ago in chordate evolution. So not even human, not even vertebrate. Chordate, which is a step above vertebrate. Anyways, the golden age of the precursor paradigm came in the 1960s and 1970s, particularly when the first evidence of a precursor of insulin was unearthed by Don Steiner and his team. Sequencing confirmed the existence of proinsulin in 1968. So these people, Don Steiner, they found a protein that looked like it had insulin and then other things associated with it. And so this is what led to the discovery of I shouldn't say the discovery, but this has led to more research going into prohormones versus hormones. Anyways, it was subsequently, pro-insulin, or the thing that comes before insulin, was shown to be relatively less active compared with insulin. So this is interesting. Even um, the pro-insulin has some activity. Now, granted, I don't know if that's working in of itself, or that if you add it, then it randomly will break apart by native proteases and have insulin activity. But regardless, this is weaker than insulin at whatever function insulin is supposed to have. So this inspiring work by Don Steiner paved the way for a much greater understanding of a whole range of pro-hormones, particularly in relation to their processing. So, as I said earlier, we stand on the shoulders of giants. Well, this Don Steiner had discovered a particular strategy for cellular signaling in one hormone. But what we can always be certain of is that if you see one strategy be employed by some cellular process, that same strategy will probably appear in many other places. So here they're saying they discovered you have proinsulin, and then that proinsulin gets proteolytically cleaved into insulin, and therefore proinsulin, this is some sort of cellular signaling metabolic pathway, if you will, that is highly regulated. Well, shoot, if this is happening with insulin, maybe it's happening with a lot of other proteins in the human body. And in this particular case, yeah, that is true. So this inspiring work and okay, in relation to the processing. So one, there are high molecular weight forms of ACTH and beta lipotropin. So although adrenocorticotropic hormone, ACTH, also called corticotropin, and beta lipotropin, beta LPH, these had been characterized separately. The concept that they were produced as part of a common precursor had not been considered, and only emerged after a number of different approaches suggested the sequences for these different peptides in the same molecule. So elegant studies by Yalow and Burson using normal human pituitary extracts and an ectopic ACTH producing thymoma. Is a tumor originating from epithelial cells of the thymus. So they took a thymoma, which is a cancer from the epithelial cells of the thymus, and they took one of those that produces ACTH, and they used that as a model cell culture to run their experiments. And with their experiments, they took human pituitary extracts. So they probably took pituitary glands from cadavers, blended them up, and then separated the components from them in order to run tests on different aliquots of those separation techniques. Anyways, these high molecular weight forms of ACTH, this indicated that ACTH was present in a high molecular weight form. So, okay, these high molecular weight forms of ACTH were also identified in the mouse pituitary tumor cell line ATT20. So they took these pituitary extracts, and I assume they separate them out by size, and some of the large-sized proteins had some of the activity of ACTH, but ACTH is a much smaller peptide. So what they concluded was there was a larger version of ACTH that was within the pituitary extracts, of the human pituitary extracts. And then they were also these same high molecular weight, or I shouldn't say exactly the same, but homologs of these were also identified in the mouse pituitary tumor cell line, ATT20. So, Lori and others went on to use human pituitary extracts and precipitated a single pro-hormone using antibodies to the different peptides. So this is pretty brilliant. So we have antibodies against, or we can buy or produce antibodies against the different peptides. And so when they used those antibodies, they didn't just take out the peptides. Multiple different antibodies took out the same single pro-hormone. So this tells you that this single pro-hormone had amino acid sequences that were associated with those individual peptides. And so what can you conclude from that? You can conclude, or you could reason, that all of those amino acid sequences were a part of the same large protein. So you could, yeah. Continuing, this was made possible because previous work by, I can't pronounce that, Shretian and Lee had discovered that gamma lipotropin sequence was found within beta lipotropin, and that it has the beta melanocyte stimulating hormone sequence at its carboxy terminal. So this is where things might be getting a little confusing, but beta lipotropin is a big peptide, but even within beta lipotropin, there is gamma lipotropin within beta lipotropin, and that, and then the beta melanocyte stimulating hormone sequence was even within that. So there are a lot of things that are holed up or contained within each other. So here, okay, so yeah, we'll just start with this. So here, processing in the anterior lobe of the pituitary in humans. So here, we have our large pro-opiomelanocortin protein. And then there is an enzyme here that can split it up into pro-ACTH and beta lipotropin. Okay, so here we have beta lipotropin and here we have pro-ACTH. And then here, pro-ACTH can be proteolytically cleaved into ACTH, or corticotropin, JP, and then NPP is the name of this, or this is I think this is NPP and also pro-gamma melanocyte stimulating hormone. Yeah, basically NP, I believe that might, regardless, this is another protein. It's hard to know all of them. So this is what happens in the pituitary gland, where you end up with these 4 different end products, if you will. Here in the hypothalamus, you start off with the pro-opiomelanocortin. It gets cleaved into this beta lipotropin and the pro-ACTH, just like what's happening up here. And then it can also get cleaved again into these 4 constituent peptides. So these are the same as up here, but in the hypothalamus, more happens. So here, this NPOMC and the pro-gamma MSH can get split into gamma-3 melanocyte stimulating hormone, and apparently it can get cleaved again into the final gamma melanocyte stimulating hormone. And here, ACTH can get broken up into ACTH 1 to 17 and clip. Wait, really? 1 second. Okay, so this is a different clip, but I will get back to that later because that looks like, yeah, so that's completely different. So just know that this is another protein or another peptide hormone. And then ACTH 1 to 17 can get processed, and then after processing can get broken down into alpha melanocyte stimulating hormone. And here gamma lipotropin can get clipped into beta melanocyte stimulating hormone. So here you can see that with one protein, depending on where you cut it up, you can get all of these other amino acid sequences that are associated with a protein, or associated with the peptide, or are the peptide, I should say. So, continuing, in 1978, the concept that POMC was a prohormone for ACTH and beta-lipotropin, this was confirmed in studies with the ACTH secreting ATT20 cell lines, which I believe are the mouse cell lines, and mains and IPER, radiolabeled amino acids in the cells, and then used immunoprecipitation and SDS gel electrophoresis. This is just a mechanism by which you can separate proteins from each other. So SDS is a sodium dodecyl sulfate. So this is a detergent that breaks down the protein structure so that all proteins lose their secondary, tertiary, and quaternary structure. Basically makes them a lot more linear. And then they use gel electrophoresis. So they put, they have the broken down or the solubilized detergent, or how to phrase that. They take the proteins that have this detergent coating them. They all have a charge on them due to the SDS, and then this, with a electric field, the electric field will push those ions or push those proteins through a gel, and then they will separate within that gel. That's the best way I can describe it. So immunoprecipitation and SDS gel electrophoresis is what's separating these radiolabeled amino acid sequences. And this is enabling them to identify a 31 kilodalton peptide, which is recognized by antibodies to both ACTH and beta-lipotropin. So they were able to discover a peptide that was about 31 kilodaltons, which is just a measurement of size, and then this large protein was could be attacked by both ACTH and beta-lipotropin attacking antibodies. So we would presume that ACTH and beta-lipotropin are both within this 31 kilodalton peptide. Anyways, Roberts and Herbert utilized a similar approach, but with cell-free translation and anti-sera to both peptides, and they reported similar results. So there's just more experimentation done to verify all of this. So I think I'll do one more, and then we will head on out for the day. So here, the emergence of the full structure of POMC. So not long after these studies, the precursor peptide was purified from rat pituitaries, and rather strangely from camel pituitaries. I wonder why they said rather strangely. Maybe it's just because it's an interesting organism. It's not a normal model organism. That might be why they said that. So Michael Tretian and Nabil Saida, I apologize for being tone deaf, I can't pronounce these names, and co-workers, then named the precursor Pro-Opiomelanocortin to reflect the known roles of the peptides in the precursors. So here, Pro-Opio, so there's an opiate, there's an endorphin in it, melano, melanin production, the melanocytes, and cortin, so the ACTH hormone or peptide hormone as well. Those are all contained with it, and that's why it's called POMC or POMC. So the same year, cloning cDNA from pituitary pars intermedia, which remember that's a part or a portion within the pituitary in between the anterior and posterior lobes, this provided the gene sequence for bovine POMC, which was independently confirmed by protein sequencing. So we didn't just do it for humans, we did it for animals as well. So similar approaches identified the sequences for the human, mouse, rat, and pig genes. So despite the sequence being highly conserved, so this is a highly conserved protein. So this could tell us that whatever function it serves in the body is extremely important among a lot of different creatures. But what that can also tell you is that this is something that links us in a way that makes us very similar. And since it makes us similar, if we have, say, a pathogen or a virus that modulates the activity of POMC in one creature, it will probably do something very similar in a different creature, because POMC is going to be expressed in both of them, and they're going to be filling similar roles in both of them. So there is some variation in the lengths of some of the peptides in different species. So this led to confusion when numbering the amino acids from the N-terminal or POMC, as the amino acids in the smaller peptides were given different nomenclatures depending on the species. So this is one problem with aligning sequences sometimes, is sometimes there are insertions and deletions in genes when you go between species. And so a lot of times we like to number the amino acid sequences so that it becomes easy to align them together. 1 second. One out. So, and because of this, sometimes the POMCs were called something completely different in different species if they were shorter than normal. Nevertheless, the structure of the gene itself is well conserved, especially in the regions covering the biologically active peptide. This is including ACTH, alpha melanocyte stimulating hormone, and beta endorphin. So the portions that are associated with actually producing the relevant peptides, those are extremely conserved, more so than the filler DNA and the filler peptides, if you will. So, importantly, there are a few key species differences that affect the processing, and this is covered in section 2E after the details of processing has been described. So here we have the POMC gene, because remember, you start off as DNA, and then the DNA gets turned into RNA, and so you can already see here, as it gets transcribed into RNA, the introns, which they don't have labeled, these introns are removed from the gene during processing into mRNA. So here we have mRNA, and so here they have a blue part and the not blue part. The blue part is associated with the production of the peptide hormones. The other parts are probably used for signaling of some form, but these parts are extremely conserved in all organisms. So this blue part is conserved. But all these other parts, so for instance, in the DNA, you could have significantly more changes in the introns here, where they have where they're using the double helix to signify it, and then also within these exons in the yellow parts, you could theoretically have a little bit more change there, where it's a little bit less conserved. So then you go from the mRNA, which this gets translated into your final protein. So here we have the human POMC, and here you can see there are different cleavage sites, and a lot of these cleavage sites are dibasic cleavage sites. So here we have an arginine and a lysine. You don't need to know too much about them, just know that they are positively charged amino acids. And so a lot of proteases will look for these positively charged ones. So, and I'll keep on going just so that we are aware. So here we have the different processing locations, and you can see in humans you have all of these different peptides that can come from the original POMC molecule. In mice, it is actually a little bit smaller, And it looks like they might actually be missing a few. Um, Maybe, yeah, they might be missing a few of those peptides, but it is a little bit smaller. And here in dogs, dogs are pretty much the same size as humans, and it looks like they produce a homologue for each human um, peptide. So with this, I think I'm going to leave because I need to go and do more stuff. But just so you know, this is a important protein that is cleaved in different sites to produce a lot of different peptides, and this has existed in the chordate kingdom, or the chordate class of animals, for probably 500 million years. So it's had 500 million years to exist and to basically fit into its role in the creature. And it's obviously not vestigial, and it's not useless because it is conserved and active in a lot of different creatures. So with that, oh, and I should also mention that these are also taking advantage of proteases, especially dibasic proteases. And in this one, this lysine, lysine, arginine, arginine, this would start to appear very similar to a furin or furin-like cleavage site as well. So keep in mind, these animals are using pro-opiomelanocortin molecules with all these cleavage sites. What else starts off as a large protein that has a bunch of cleavage sites on it to get into their final functional forms? Viruses. Virus proteins very frequently utilize this exact same strategy. So if you think with me just a little bit, if you bear with me, I can't find a paper on this, but think about this with me. If you're a virus and you know or you utilize proteases already to get into your active forms, why not use the same proteases that are used for this peptide processing? And another thing to think about is, why don't you not only use those same proteins, but why don't you even mimic the proteins that why don't you even mimic those same peptides? Because then, if they try to make an antibody against your proteins, well, they're going to cross-react with these proteins as well. So this is my presumption that is happening, because we know already that for influenza and coronaviruses, these two subsets of viruses often mimic ACTH. This is a peptide hormone that is mimicked by viruses. And then when your body makes antibodies against those viruses, they will often attack ACTH. And when they attack ACTH, what do you think happens? You end up not being able to release cortisol as much. And then when you're not able to release cortisol, you end up becoming more inflamed, and then you become leakier, and you become more symptomatic, and therefore you become more contagious. So you help the virus spread that way. And if you look here, ACTH has a cleavage site associated with it, and this actually, I did a sequence alignment of POMC with the spike protein of SARS-CoV-2, and it lined up pretty well. One of the cleavage sites of the spike protein lined up pretty well with this. And I believe the other cleavage site, the S2' cleavage site, lined up pretty well with, where is it? beta endorphin, I believe is what it was. So I believe it lined up pretty well here. So what else do I want to talk about? I think that's sufficient. Just I personally, this is my philosophical view on all of it right now. I believe that viruses specifically use this enzyme. I believe they, or not this enzyme, I believe that they specifically target this protein. Now, granted, maybe it was viruses facilitated in the insertion of ACTH or of something within the opiate or within the beta endorphin gene, but I'd like to think that if we have this really highly conserved peptide hormone, or I should say pro-peptide hormone, that's been around in pretty much all chordates for 500 million years, then viruses are aware of it and will end up utilizing the commonalities between their victims. So I'd like to think that these viruses might be co-evolving with POMC, at least to a degree. And we already know that some viruses do mimic ACTH, and they mimic it. There's a lot of weird theories as to how it mimics ACTH, but I like to think it's as simple as it's basically trying to imitate this entire pro-opiomelanocortin protein, and by doing so, it is able to screw up all of these other downstream peptides. So that is my current hypothesis right now. I would like it if some research would go into that, but obviously I don't have a lab myself, so I can't. But yeah, I think that's interesting. And if that is true, then all these viruses that imitate this pro-opiomelanocortin protein, as they do it, they might cause things such as Alzheimer's due to initiating autoimmunity. They might cause multiple sclerosis. They might be causing all of these other diseases that are the result of autoantibodies. And if these autoantibodies are arising or getting upregulated because these viruses are mimicking very important proteins in the body, right there you just have a cause for a lot of really debilitating diseases of old age. So with that, I'd like to shut this off, and hopefully I didn't ramble too much, but I will see you tomorrow.